Amyloids are big and ugly and easy to spot under a microscope, so they've gotten most of the attention. But our research shows they're just the tip of the iceberg. That's the stunning assessment from Dr. Stephen Fried, a lead researcher at Johns Hopkins University, and it's a statement that could send shockwaves through the entire field of neuroscience.
For decades, the global war on Alzheimer's disease has been fought on a very specific battlefield against two well-known enemies, clumps of protein called amyloid and tau. They were the villains, the primary culprits behind the devastating memory loss that afflicts millions. But what if this entire time we've only been seeing a fraction of the real enemy?
Today, we're diving deep into a bombshell breakthrough that suggests Alzheimer's is far more complex and far more insidious than we ever imagined. In this investigation, we'll uncover what scientists at Johns Hopkins have just discovered inside the brain. We will explore how this new finding completely turns our understanding of Alzheimer's on its head, revealing potentially hundreds of hidden culprits that have gone undetected until now.
And we will ask the most critical question of all. Does this discovery represent a true game changer, a pivotal moment that could finally set us on the path to curing this heartbreaking disease? To understand the magnitude of this breakthrough, we first need to look at the story we've been told about Alzheimer's for generations. It's a story of plaques and tangles.
Imagine the intricate, high-speed network of your brain, where billions of neurons fire off signals every microsecond, creating your thoughts, memories, and personality. Now, picture that network being slowly clogged and choked by a toxic buildup. That's the classic picture of Alzheimer's. The first villain is a protein called amyloid beta. In a healthy brain, these protein fragments are cleared away. But in an Alzheimer's patient, they misfold and stick together, forming hard, insoluble clumps known as amyloid plaques. The second villain is tau. Tau proteins are supposed to be like the railroad tracks that help transport essential nutrients and materials within a brain cell. In Alzheimer's, these tracks twist and collapse into what are called neurofibrillary tangles, causing the cell's internal transport system to grind to a halt, leading to cell death. This two-pronged attack, formalized as the amyloid cascade hypothesis, has been the guiding star of Alzheimer's research for more than 30 years. It proposed a linear sequence of events. Amyloid plaques form first, which then triggers the formation of tau tangles, leading to widespread neuronal death. It was a clean, compelling story, and it channeled billions of dollars and the careers of thousands of scientists into a singular mission. Stop amyloid, and you stop the disease. But this laser focus came with a growing sense of unease. Drug trial after drug trial, designed to clear these plaques, ended in failure. Some drugs effectively removed the amyloid from the brain, yet patients' cognitive decline continued unabated. This led to a nagging, whispered question in the halls of neuroscience conferences. Are we missing something? Is the big and ugly amyloid plaque a cause of the disease, or merely a tombstone marking where a cell has already died from some other cause? The stakes could not be higher. In the United States alone, more than 7.2 million adults over the age of 65 are currently living with Alzheimer's.
Over 100,000 people die from it each and every year, and the Alzheimer's Association delivers a chilling warning. By the year 2050, that number could soar to nearly 13 million Americans. Patients progressively lose their memories, their ability to speak, to care for themselves, and eventually even to respond to the world around them. It's a cruel, slow erasure of the self, and for decades, we thought we knew the enemy.
But what if the plaques and tangles were just the most obvious criminals, the ones leaving the biggest, messiest evidence at the scene, while a whole gang of other saboteurs operated in the shadows? This is where the team at Johns Hopkins University enters the story, with a clever and revealing experiment. The study, published in the prestigious journal Science Advances, was conducted not on humans, but on 17 elderly rats, all 2 years old, and raised in the exact same pristine laboratory conditions. The researchers put these aging animals through a series of rigorous memory and problem-solving tasks, essentially giving them cognitive checkups. These weren't simple mazes. Think of complex tasks designed to test what's known as spatial learning and memory, the very same functions that are cruelly stripped away in early human Alzheimer's. Tasks like remembering the location of a hidden platform in a pool of water, a test that requires a healthy hippocampus to navigate effectively. The brilliance of this setup was its realism. These weren't rats genetically engineered to overproduce human amyloid protein, a common but artificial model. These were normal rats living out their full lifespan, with some naturally developing the rodent equivalent of senior moments and others remaining sharp as a tack. This allowed the scientists to ask a more profound question. In a brain that is aging naturally, what is the true molecular signature of cognitive failure? The results were startling. Despite their identical upbringing and age, 7 of the rats showed clear signs of cognitive impairment. The other 10 rats, however, performed brilliantly. In fact, their scores were just as good as those of healthy 6-month-old rats. This created a perfect, naturally occurring control group. The scientists now had 2 sets of brains to compare, those that were aging gracefully and those that were succumbing to cognitive decline. They focused their attention on the hippocampus, the seahorse-shaped region of the brain that is absolutely critical for learning and memory, and one of the first areas to be ravaged by Alzheimer's. And what they found there was the bombshell. In the brains of the 7 cognitively impaired rats, they discovered that over 200 different proteins were misfolded. Let me repeat that, over 200. And these were not the usual suspects. This was a vast, previously unknown collection of misshapen proteins, all potentially gumming up the works of the brain. In the 10 healthy rats, those same proteins had maintained their correct functional shapes. The conclusion was undeniable. The cognitive decline wasn't just about amyloid and tau. A whole host of other proteins were contributing to the damage. So what exactly is a misfolded protein, and why is it so dangerous? Think of a protein as a tiny, biological machine designed to do a specific job. For it to work, it has to be folded into a precise 3-dimensional shape, like a key that has to be cut just right to fit a lock. If the protein folds into the wrong shape, if the key is bent, it can't do its job. Worse, these improperly shaped proteins can become sticky and interfere with other cellular processes, causing widespread dysfunction and damage. This is precisely what happens with amyloid and tau. They misfold and clump together. But the revolutionary finding here is the sheer scale of the problem. Lead researcher Dr. Stephen Fried explained why these 200 other culprits have remained hidden in plain sight for so long. As he put it, amyloids are big and ugly and easy to spot under a microscope, so they've gotten most of the attention. They form massive, obvious plaques. These other 200-plus proteins, however, don't necessarily form large clumps. They are more like individual agents of chaos, subtly disrupting brain function without creating the large, easily detectable aggregates that researchers have been trained to look for. This is what Dr. Fried meant when he said they were just the tip of the iceberg. We've been focusing on the visible part of the problem, while a massive, hidden network of dysfunction lay submerged beneath the surface. He added, "We think there are a lot of proteins that can be misfolded, not form amyloids, and still be problematic." This single study potentially rewrites decades of Alzheimer's research, suggesting the problem is exponentially more complex than we believed. It's a paradigm shift in our understanding of this disease. If you find this kind of scientific detective work as fascinating as we do, and believe this knowledge is crucial, take a quick moment to hit that like button. It truly helps more people discover this important information. Now, for the crucial dose of reality. This groundbreaking research was done on rats. As we all know, a finding in an animal model, no matter how promising, is not the same as a proven fact in humans. This is where the perspective of other experts becomes vital. Dr. Keith Vossel, a professor of neurology at UCLA, weighed in on the study, calling it an incremental advance, but it definitely builds on our knowledge base about this disease. He provides a necessary note of caution, but also acknowledges the study's importance. So how do we bridge the gap from rats to people? Dr. Vossel lays out the roadmap. He states that confirming this in humans would require autopsy-based studies or examining surgically resected hippocampal tissue from patients. He explains, that's really the only way to assess it directly. In other words, scientists need to get access to human brain tissue and perform the same kind of deep protein analysis that the Johns Hopkins team did on the rats. Until then, the link remains a very strong and compelling association, but not yet definitive proof of causation in humans. As Dr. Vossel summarized, at the moment it's a nice association, it just requires a lot more follow-up. Despite the need for more research, the implications for the future of Alzheimer's treatment are staggering. For years, the pharmaceutical pipeline has been filled with drugs that target amyloid and tau, with many high-profile and heartbreaking failures. This new discovery could explain why. If you're only targeting 2 culprits out of more than 200, you might only be chipping away at the problem, not solving it. This finding opens the door to a completely new therapeutic landscape. This idea might sound familiar. It mirrors one of the greatest medical success stories of our time, the fight against HIV/AIDS. In the 1980s, HIV was an unstoppable death sentence. The breakthrough came with a cocktail of drugs that attack the virus from multiple angles simultaneously. It turned a fatal diagnosis into a manageable chronic condition. Could we be on the cusp of a similar moment for Alzheimer's? Instead of one silver bullet drug, the future might be a personalized neurococktail. One drug to support cellular energy, another to help proteins fold correctly, and perhaps a third to target the classic plaques and tangles. This discovery of 200 new targets makes that once fantastical idea a tangible possibility. Furthermore, the implications extend beyond treatment to diagnosis. If these misfolded proteins can be identified in cerebrospinal fluid or blood, they could become powerful biomarkers, an early warning system for the brain, flagging the disease years before the first memory fades. The research isn't just an abstract scientific curiosity, it's deeply personal. As Dr. Fried himself noted, a lot of us have experienced a loved one or a relative who has become less capable of doing those everyday tasks that require cognitive abilities. This quest for knowledge is driven by a desire to prevent that suffering. Understanding what's physically going on in the brain, he says, could lead to better treatments and preventative measures. The next step for his team is to take this discovery to the next level of detail. They plan to use high-resolution microscopes to zoom in on these newly identified rogue proteins to see their deformed shapes at the molecular level. By understanding their exact structure, they can begin to design ways to fix them or clear them out. So, what is the ultimate takeaway from this bombshell Alzheimer's breakthrough? It's that the enemy is bigger, more complex, and more cunning than we ever knew. The iconic villains, amyloid plaques and tau tangles, are not the whole story. They may just be the big and ugly tip of a vast iceberg of cellular chaos. This discovery from Johns Hopkins doesn't offer an immediate cure, but it does offer something almost as valuable, a new map. A map that points to over 200 previously hidden targets in the war against cognitive decline. The road ahead is long. It will require painstaking work, human studies, and brilliant new drug design. But for the first time in a long time, it feels like we are finally looking at the whole battlefield. The fight is far from over, but with this new intelligence, our chances of winning have just gotten significantly better. To follow every step of this journey and to stay informed on other critical breakthroughs in science that could change our world, make sure you are subscribed and have notifications turned on. You truly won't want to miss what comes next in this incredible story.